Hello and good morning, ladies and gentlemen. I am Ashutosh Raj, and today I am going to deliver my topic, that is oral delivery of amphotericin B-loaded nanopolymersomes on the occasion of second international conference on women in multifaceted research. So this will be the flow presentation in which firstly I will talk about introduction, later objective, a specific aims, our experimental work, and last I will conclude my slide. So let's talk about amphotericin B-loaded polymersomes. You must have heard about leishmaniasis, kala-jar, or black fever. Leishmaniasis is a protozoal disease caused by different species of leishmania and transmitted through the infective bite of 70 different species of an insect vector, that is the plipitomyine. Leishmaniasis is an endemic disease in more than 60 countries worldwide, including South Europe, North Africa, Middle East, Central and South America, and the Indian subcontinent. It has been reported that more than 90% of the global total of leishmaniasis cases occur in 5 developing countries across 3 continents, that is northeastern India, Bangladesh and Nepal in Indian subcontinent, Sudan in Africa, and northeastern Brazil. Recently, the number of reported cases in different geographical areas have increased with 5 lakh annually and 12 million people are infected globally. Out of this, 80,000 people lost their life due to untreated leishmaniasis. Untreated visceral leishmaniasis carries a mortality rate of 75 to 95%, while cutaneous leishmaniasis can spread to skin, involves the mucosa, resulting in death from secondary infection. So we have formulated the amphotericin B. So US FDA has given a dosage regimen having drug like pentavalent antimony, paramomycin sulfate, pentamidine isethionate, and the amphotericin B. Amphotericin B is a polyene macrolide BCS class 4 drug, that is amphiphilic antibiotic. It has a broad spectrum of activity with no major problem of resistance. Different marketed formulations are available for leishmaniasis treatment like fungizome, ambizone, amblicet, ampicil, etc. All these formulations are administered intravenously, and the main problem associated with IV drugs are its stability and the toxicity. Also, due to increased cost of lipid-based formulation, this is very difficult to bring in market, and the poor people and many peoples cannot afford it. So here comes our main objective, that is to develop amphotericin B formulation intended to be administered orally with enhanced bioavailability. Here comes our specific aims, in which we will talk about analytical and bioanalytical method revalidation of amphotericin B, synthetic and characterization of a polymer, preparation and characterization of amphotericin B loaded polymersomes, that is PAMBO, and investigation of in vitro stability study of PAMBO in simulated gastrointestinal fluids. And at last, we will talk about intraduodenal pharmacokinetic of amphotericin B loaded polymersomes formulations. Here it comes our experimental work, in which first of all we synthesize and characterize the monomethoxy polyethylene glycol citrate PL block copolymer, and in second part we did a HPLC analytical method revalidation and preparation and characterization of amphotericin B loaded polymersomes, that is PAMBO, and at last we did an intraduodenal pharmacokinetic of amphotericin B loaded formulation. So here comes what first, that is synthesis of monomethoxy polyethylene glycol citrate PL block copolymer. Copolymer from monomethoxy polyethylene glycol and PLA were already synthesized in our lab. It is synthesized by ring opening polymerization using citric acid as a linker, and synthesis was carried out in two steps. That is, in first step, we did a synthesis of monomethoxy polyethylene glycol citrate. For synthesis of PGC citrate acid first converted to citric acid chloride, a mixture of anhydrous citric acid and thionyl chloride. It was refluxed in 30 ml of dry toluene at 120 degrees centigrade for 24 hours. Separately, monomethoxy polyethylene glycol was dissolved in tetrahydrofuran in 15 ml THF, and the solution was dried in tetrahydrofuran, was added dropwise to the reaction mixture and kept under stirring for 30 minutes in ice bath. And purification of crude product was done by column chromatography using 1:1 mixture of ethyl acetate and methanol as a eluent. And at last, purified product was characterized using aqueous gel permeation chromatography, that is GPC. After that, we did a synthesis of monomethoxy polyethylene glycol citrate polydilactic acid. For a particular batch size of block polymer synthesis, a fixed molar ratio of PGC and dilactide were melted at 120 degrees centigrade under vacuum and stirred for 2 hours. After that, a stannous octanate solution in dry toluene was added under nitrogen atmosphere and temperature was raised to 150 degrees centigrade. Polymerization reaction was continued for 22 hours. After polymerization, reaction mixture was allowed to cool and polymer was dissolved in dichloromethane and purified by precipitation in ice cold. That is 1:1 mixture of diethyl acetate, ether and petroleum ether. Precipitate was collected, dried and stored at minus 20 degrees centigrade for further use. The scheme of polymer synthesis is given. Now, we did a characterization of monomethoxy polyethylene glycol citrate PL block copolymer. For the characterization, we did a FTIR for the citric acid chloride, that is Fourier transform infrared spectroscopy. For FTIR for citric acid chloride, we know that citric acid, the affinity or the potency of citric acid was increased by thionyl chloride. So you must see the peak at 1792.08. This peak and another peak obtained at 782.45. So this indicates the citric acid chloride. Now FTIR for PgCl. For PgCl FTIR, different peak obtained, that is 2997, 1760, 1090 and 752. So this type, if you take a look about GPC peak of PGC, that is gel permeation chromatography peak of polyethylene glycol, you see a different peak of graph. So this different peak of graph indicates the different retention time. If you talk about monomethoxy polyethylene glycol of molecular weight 2000, its retention time obtained at 10.95 minutes, and for monomethoxy polyethylene glycol at molecular weight 1200, it obtains at 11.20, and for PGC it is 10.05 minutes. So this indicates a different elution time. Also, if you talk about protonated nucleomagnetic resonance NMR, it indicates the percentage of hydrophilic portion, and NMR spectra of the polymer was done for molecular weight determination, confirmation, and also the percentage of hydrophilic portion in the block polymer. Later, we did a HPLC analytical method revalidation. For HPLC analytical method revalidation, we have taken a mobile phase of acetonitrile and disodium ethylene diamine dihydrate, and the ratio of 42:58. Now we has taken a column of inert seal ODS 3B, and we have taken an injection ball of 10 microliter at a flow rate of 1 ml per minute and at temperature of 30 degrees centigrade, and period detector detect it at the wavelength of 408 nanometer. And in the later phase, we did a chromatographic condition for bioanalytical determination of amphotericin B. In the bioanalytical determination of amphotericin B, we has taken also the same mobile phase, and the column was same, and the flow rate is 1 ml per minute, but here the injection volume is 50 microliter, and also it is detected by PDA at the wavelength of 408 nanometer, and at its running time is 70 minutes. If you take a look of HPLC chromatogram of amphotericin B, okay, HPLC chromatograph of amphotericin B, so this indicates that the amphotericin B has a sharp curve. Okay, this amphotericin B has a sharp curve, and it indicates the drug substance have been eluted in a single symmetric and a pure peaks. Accuracy and precision were determined from QC sample at 3 different concentration in the calibration range, that is in triplets. And also this table, B table suggested that B several stated that method is sufficiently accurate and precise, and sensitivity of the method was determined by limit of quantification, which is the minimum quantifiable concentration, and limit of quantification is calculated by the mathematical equations. I am not going there. So if you talk about the different HPLC, also HPLC method was found to be suitable for analysis of in vitro samples. Okay, accuracy and precision for HPLC analytical method was within limit of ICH guidelines, that is International Convention of Harmonization. It in within limit means it occurs within the limits of ICH guidelines. If you take a look at second experiment, at different amphotericin B has a retention time of 4.8 minute, and the second one, that is the internal standard, it has a retention time of 11.02 minutes, 2 minutes, and calibration curve was plotted with 6 concentration over the range of 20 to 2000 nanogram per ml by spiking known concentration of drug in plasma, blank plasma. Okay, and if you talk about lower limit of quantification, that lower limit of quantification occur at 20 nanogram per ml, and we all know that the lower limit of quantification is defined as a total concentration on the calibration curve that could be measured, okay, with acceptable precision and accuracy. Okay, this will be taken in the limit of precision and the accuracy. Now we prepare amphotericin B loaded polymersomes, that is the PAMBO. Okay, we also prepared PAMBO with the help of 60 mg of polymer. 60 mg of polymer was and also we know that a PAMBO was prepared by solvent injection method. 60 mg of polymer of polyethylene glycol citrate lactic acid and 4 mg of amphotericin B was dissolved in 200 microliter of DMSO, that is dimethyl sulfoxide, which was common solvent for both molecules, followed by 2 ml of ethyl acetate was added, and the solution was added to 5 ml of aqueous surfactant solution, that is 1% PVA in phosphate buffer, with a constant stirring up to 30 minutes at 45 degrees centigrade. Okay, then dispersion was processed. Okay, then dispersion was processed for size reduction. Size reduction was done by, okay, this is question for you, size reduction was done by homogenizer, okay, at the 25,000 gram for 7 minutes. After that, dispersion was added to 50 ml of water and again stirred until ethyl acetate subsided. You just look at this flow chart here, everything is mentioned, that 50 ml of water and again stirred until ethyl acetate subsided. Now we take a look at stability of amphotericin B drug loaded PAMBO in simulated gastrointestinal fluid. A stability under GIT condition is, we all know that stability is important for in GIT condition, because we eat a food, either it will be accumulate at a single place, or it is necessary to move or metabolize inside the body. So this stability study is important to be conducted. Okay, the incorporation of PAMBO in intraduodenal delivery was explored as a method to enhance the oral bioavailability. So pharmacokinetic studies were also were conducted, and you just look at that particle size first, initials for simulated gastric fluid and simulated intestinal fluid, faded state simulated intestinal fluid, and fasted state simulated intestinal fluid. So the conclusion of overall slide was that it can be observed from table that polymersomes is not stable in simulated gastric fluid. We observed that amphotericin B polymersomes is not stable in simulated gastric fluid, whereas in simulated intestinal fluid fed in fasted state, it is stable up to 6 hours, and no significant changes were observed in the particle size, polydispersity index, and entrapment efficiency of PAMBO. And this confirms the stability of PAMBO in simulated intestinal fluid. So we just concluded from this slide that our PAMBO is stable in intestine, that is in the duodenal region of the rat. We just dissect it, and after injecting our drug in the duodenum of the rat, we found it is stable. So the formulation was just be intrigue coated. Our formulation must be intrigue coated to pass the gastric fluid because our gastric fluid make it unstable and toxic. So we did a characterization of amphotericin B loaded polymerism, that is the PAMBO. So here take a great curve obtained, that is amphotericin PAMBO has a size of 274.01 nanometer with a polydispersivity index of 0.23. If you take a look at PXRD of different formulation, that is the amphotericin B loaded form polymerisms, blank polymerisms, physical mixture. Okay, pure amphotericin B, you must take a look that the halo pattern was observed for amphotericin B loaded polymerism. So this halo pattern indicates its morphism, that it is amorphous in nature. Okay, this graph, so it's amorphism. Also, pure amphotericin B is slightly less amorphous than our PAMBO, and it has a more inclination towards the crystalline forms, right? So if you take a look of we considered a scanning electron microscopy. In a scanning electron microscopy, each bright image of polymerisms were obtained with substantiated vesicle formation. This bright edge of the polymerisome was due to the formation of thick polymeric membrane. Okay, the polymerisms were spherical to oval shape with particle size both in micron and in nano range. Right. In transmission electron microscopy, you see a different transmission means diameter of 236 nanometer and different transmission. Now, if you talk about in vitro release study, we did also a in vitro release study. Stability under GIT condition is crucial for absorption of PAMBO in GIT. Since these polymerisms have been developed for oral delivery, okay. So if you take a look, we did this for 7 days. Okay, 7 days at 37 degrees centigrade at 80 rpm, 80 rpm, and we found that at first it gets releases too much. Okay, it get releases too much. Polymerisms get releases too much and after some time, to further mass balance, after 5 days about 72% of drug was released from PAMBO, and this so that it is long acting and can easily we can easily administer orally. Now at last, that is intraduodenal study of pharmacokinetic of amphotericin B loaded formulation wants, right? From this graph, we have taken a different marketed formulation, that is the PAMBO, free drug suspension, ambisome, fungizome, and we found that the different formulation occurs, so different plasma concentration and at different time. And after the conclusion of all the different drug, we find our PAMBO has a maximum concentration at a maximum time with area under curve total of 11,671.06 with a T half means half life of 20.38. So we did this in the drug in the SD rat at 5 mg per kg with a different dose. So let's conclude all the slide, and our conclusion of overall slide is that amphotericin B encapsulated polymerisms, that is PAMBO, were formulated and characterized in vitro for size range, PDI, zeta potential, surface morphology, encapsulation efficiency, solid state characterization of API, and drug release profile. Results depicted spherical morphology of PAMBO having average number size of 274 nanometer with about 40% encapsulation efficiency. Approximately 70% of encapsulated drug was released from PAMBO over a period of 7 days in vitro drug release study. PXRD study reveals amorphous nature of drug in the formulation. GI stability result depicted that PAMBO were not stable in simulated gastric fluid. Hence, for oral delivery of formulation, it was postulated to deliver in coated capsule. Okay, we just talked about its stability and toxicity, right? And at last, we concluded that intraduodenal pharmacokinetics in rats showed promising results. Precisely 14.41 and 20.80 and 4.75 times increases in bioavailability of amphotericin B was observed in PAMBO with respect to free drug, fungizome and ambisome respectively. So with this conclusion, we have found that our PAMBO, that is polymersome amphotericin B, is much more can be given by oral route and it is stable in the duodenum region. Okay, so thank you so much for hearing me, and if you have any query or question, you can put it in a comment box and I will reply it as soon as possible. So thank you so much.